Hello everyone, I welcome you all to my YouTube channel, the Future Synthesis Academy. First of all, I would like to thank everyone who is viewing and who is basically commenting on my video. And at the end of this video, I will be asking question related to the previous video. So you people need to watch the previous video to answer the question. And today's topic is seen that it's endosymbiotic theory. तो इसे आज आरंभ से प्रारंभ करते हैं and let's know what is this. So this endosymbiotic theory basically sets the vitality for existence of certain cellular organelle. Now we know that photosynthesis basically happens in chloroplast, and respiration is basically related with mitochondria. So endosymbiotic theory is considered as one of the greatest achievement of 20th century evolutionary biology. Now the foundation stone was basically laid in 1905 by a Russian botanist who was known as Konstantin Merezkovsky. Basically, Konstantin Merezkovsky was working with lichens, and we know that lichens lives in a symbiotic association. He was working with lichens, and lichens lives in a symbiotic association with algae and fungi. Now the entire basis of his experiment was dealing with the influence of work of another scientist, and this association basically he called it as symbiosis. So basically the foundation of his experiment was basically led by the work of another scientist who is known as Andreas Franz Wilhelm Schempmer. Now, Andreas Franz Wilhelm Schempmer, he knew that that there is some kind of relationship between cyanobacteria and chloroplast, whereas Konstantin Merezkovsky was basically thinking that large, more complex eukaryotic cells evolved from symbiotic association between less complex ones. So after this, there was another great scientist who came into the recognition. She was Lynn Margulis. Lynn Margulis was basically a very staunch and primary modern proponent of laying the foundation stone of endosymbiotic theory. And Lynn believed that there were two endosymbiotic events that took place, and she had this idea that there was some kind of symbiosis involved in the evolution of eukaryotes, and she basically championed it. She was one of the best known American evolutionary biologists. Now, the existence of the eukaryotic cell was basically that Lin believed that mitochondria and chloroplast were once independent bacteria. This was the thought which was led into the scientific world, in front of the scientific world, by Lynn Margulis. And then there was another scientist who came into the explaining this endosymbiotic theory, was Wilhelm Ziegler. Now, Wolfram Zelig was a German biochemist, and he was a believer that there was some kind of fusion event happened between the ancestor of archaea and ancestor of bacteria, and because of this fusion event, the eukaryotic organism came into this world. So these were the four major scientists who really contributed in development of the endosymbiotic theory. It started in 1908, and ultimately this theory was accepted in till 1980. Everything regarding this theory was basically proven with experiments. So in today's video, I will be basically discussing the endosymbiotic theory. The scientists are these who led the foundation, and I will be also dealing with the diagrammatic representation with the endosymbiotic theory. So what happened was there was a cell which is known as a progenote. So we cannot say it's cell because this is a hypothetical stage which happened in evolutionary biology. So progenotes are basically termed as the last universal common ancestor. So due to certain geographical isolation, this progenote was basically divided into two parts. One one became pre-bacteria, pre-bacterium, and another became a pre-archean. And due to this geographical isolation, they developed their own genetic mechanism, and their own metabolic pathways. So this progenote was basically at one point of time was permeable, and this permeability was really important because the progenote was permeable, which led to sharing of certain genetic information. Now, what happened that this pre-bacterium started developing its own cellular membrane, which was basically a yeast cell membrane. And this pre-archean started developing its own cell membrane, which was basically a ether cell membrane. Now, certain atmospheric changes and event led to a very vital step in this endosymbiotic theory, was the fusion reaction as proposed by Wolfram Zillig. This fusion happened, and this fusion event led to the development of pre-eukarya with the Easter membrane. Now, what happened that over here, Zillig was absolutely correct that a fusion event happened which led to the formation of pre-eukarya with the Easter membrane. Now, then there was certain again transformation which was happening in the formation of the earth and the formation of the cell. But at this point of time, this pre-eukarya basically developed into a eukaryote. Now, this eukaryotic cell with nucleus happened because the plasma membrane started itself folding around the chromosome and the development of the nucleus happened. Now, then there was this pre-mitochondrion which was nothing but a type of bacteria. I will be illustrating in a minute that how this was bacteria. So this bacteria started itself this bacteria was basically engulfed by endocytosis by this eukaryotic cell. Now, this is not very much evidently clear whether this pre-mitochondrion became a prey or it was a predator. There is no written proof or evidence that this engulfment happened because of certain advantage of the eukaryotic cell or certain advantage of the pre-mitochondria. So this pre-mitochondria entered inside the eukaryotic cell and started living independently. Now then, the eukaryotic cell had nucleus and this pre-mitochondrion was converted into a mitochondria. Now, this event must have happened with certain advantage of the pre-mitochondria. This pre-mitochondria had a cellular membrane because of which it was able to avoid the digestion, which was easily culminated by the eukaryotic cell, but it kind of dodged that process. Now, there was another form where this Easter membrane pre-bacteria developed alga with chloroplast. And here we are with chloroplast where photosynthesis happens. Now, this alga which had this chloroplast, it is believed that this chloroplast was nothing but a pre-chloroplast. Now, this pre-chloroplast was basically again taken inside this pre-bacterium and it started living independently. So both this pre-mitochondrion and pre-chloroplast started living independently. Now, why they started living independently sets the vitality of the existence of mitochondria and chloroplast. Now, it was seen, the genetic study was done, and it was seen that pre-mitochondrion shares certain kind of similarity, the genetic similarity with rickettsial genome, the bacteria Rickettsia, which causes the Rocky Mountain fever. And it is also alpha proteobacteria, a gram-negative bacteria, which sometimes behaves as a gram variable, having a pleomorphic nature that can change their shapes. And this pre-chloroplast was said that they were nothing but cyanobacteria. Now, it was seen that yes, the chloroplast has certain kind of similarity with the cyanobacteria. Means the cyanobacteria has the same photosynthetic reaction center, and the chloroplast also has the same photosynthetic reaction center. The chloroplast also has 70S ribosome, which the cyanobacteria also has. The cyanobacteria is basically a gram-negative bacteria. So this also has, and the rickettsial genome was basically found out by 16 RNA analysis, that this is nothing but an ancestor or descendant of Rickettsia. So the cyanobacteria and the Rickettsia basically were absolutely bacterial microorganisms which had this ability to exist independently, putting the theory, which was putting the hypothesis which was laid by Lynn-Margulis. So this cyanobacteria and this Rickettsia also share certain similarity. Now, the kind of similarity that is seen in mitochondria as well as the chloroplast is also seen in Rickettsia and cyanobacteria. For instance, the kind of porin which is found in outer membrane of cyanobacteria and Rickettsia is also found in mitochondria and chloroplast for transportation. Then there is cardiolipin, which is found in the inner membrane of the mitochondria, is also found in inner membrane of the Rickettsia. Then there is genetic element that the cyanobacteria basically has the DNA content which has been reduced in chloroplast. Basically, chloroplast DNA content is about 120 kb to 200, and the mitochondria has a DNA content of about 13 kb. The chloroplast basically synthesizes around 120 genes, whereas the mitochondria has 37 genes. So this is the whole endosymbiotic theory for the existence of the chloroplast and the mitochondria in the eukaryotic cell. So if we can find out other way of evidence that there are certain more evidence into the existence, is basically that once the mitochondria or the chloroplast is removed, the cell does not have any ability to regenerate the removed mitochondria or the chloroplast. So they have their own DNA content and they are semi-autonomous, but their vitality is also controlled by nuclear genome. And there is a reduction process which happened during the course of evolution, because the rickettsial genome is really huge, it's humongous, but it has been reduced in terms of mitochondria, and the cyanobacteria genomic content is also reduced. Basically, the cyanobacteria did one of the greatest thing, which is known as the great oxidation event, which led that the synthesized molecular oxygen was basically used to convert the reducing environment into an oxidizing environment. So it was basically the cyanobacteria. So this sets the whole vitality for the existence of the chloroplast. And this endosymbiotic theory is really important because we should know that where the chloroplast came into the existence, from where it came. So this was the whole theory on which the chloroplast established itself as an independent cell organelle and a cell inside a cell, basically. I hope that I made this really interesting. And the question is, from my previous video, the question is basically what kind of phosphorylation is basically performed during oxygenic photosynthesis and anoxygenic photosynthesis. Please do like, share and subscribe my channel. Thank you. And जहाँ रहें स्वच्छ रहें, साफ रहें और प्रकृति का ध्यान रखें. Thank you. धन्यवाद. Happy learning.
Thank you.